So thank you so much for having us. So I'm Izzy, I'm a PhD student at MFIG, and I'm going to talk to you about how we identify new drug targets to fight Aspergillus infections. So we had a bit from Paul earlier about the azoles and how they work, so I'm just going to go over this briefly. So as a lot of you will know, azoles are the primary treatment for aspergillosis, and they work by targeting the CYP51 protein, which makes ergosterol. So you can see here in purple, this is the CYP51 protein, and it makes ergosterol from the ergosterol precursor, and then this ergosterol goes into the cell membrane and makes it nice and fluid and able to work properly. And you can see on this side, when we add the azole drugs, the azole binds to the CYP51 enzyme where the ergosterol also does. So it means that it can't make this ergosterol anymore, and that makes the cell membrane unstable and leads to reduced fungal growth. So one of the problems with the azoles is there's been increase in resistance. So the fungi change their CYP51 protein in some way, which means that that the drugs can't bind anymore. So we're always on the lookout to look for new drug targets so we can make new drugs. So some of the new drugs that are currently in clinical trials for aspergillosis target lots of different enzymes, not just the CYP51 enzyme. And I'm really sorry if I butcher these names, but fosmanogepix targets the GWT1 protein. I can't see this, sorry. Resofungin targets the glucan synthase protein, and olorofin, which we heard a bit about earlier, targets the pyrimidine synthesis proteins. So targeting different fungal proteins mean that we can overcome the resistance problems with the azoles to the CYP51 protein, and it means we can attack the fungi from all angles if we're attacking different things inside the cell. So why is it difficult to find new fungal drugs? So here is a picture of us working in the lab. And our DNA makes, as Paul said, thousands of different genes, and these genes all add instructions for our bodies to make different proteins. And drugs target different proteins. And it's a bit easier for scientists to come up with new drugs to target bacterial infections, because bacteria share about 2% of their genes with us, which means that there's 98% of their genes and then 9% of their proteins, which we can make new drugs to target. However, fungi share about 50% of their genes with us, meaning that we have a lot less genes and proteins to target. So what do we look for when we're trying to come up with new antifungal drug targets? So one of the reasons that's hard to come up, as I just mentioned, with new drugs is that we share a lot of similarities with the fungi. So we need to make sure that these drugs are selective for fungal cells so they don't harm our human cells when we take these drugs. So this will lead to off-target effects and make the drugs toxic to us, so we don't want that. The genes and proteins that we do target need to be essential or important for growth. So there's no point targeting a gene or a protein which makes the fungi green, because it won't kill the fungi. So these genes need to encode something important within the fungi that when we stop it working, it kills the fungi. And the protein that we target also needs to be druggable. And this is a term that we use to describe the shape and the properties of the protein, which means that molecules can bind to it to stop it from working. So I'm just going to talk to you about a few projects that we have going on in the lab at the moment. So one of these projects is the NimT project. So mitosis is the process where one cell becomes 2, and then 2 become 4, and 4 become 8, and so on. And this is the main mechanism that fungi use to grow. So you can see here, this black dot is the nuclei, all the genetic material in the DNA is, and they divide, and so on and so on. So one of the main processes that this can happen is by interaction of these two proteins called NimX and NimT. So it's actually the physical touching of these two proteins inside the fungi that allow this mitosis and the cell cycle to happen. So we hypothesize that if we can stop these two proteins physically interacting by finding a drug that binds in between them, stops them touching, then we can stop this. We can stop the fungi growing and dividing. So how are we doing this in the lab? So first step is to make computational models of the NimT and NimX proteins. And you can see here in blue, this is a model of NimT. Once we've built these models and seen the shape we can simulate on the computer how they interact with each other, so how they're actually physically interacting inside the cell. We can then screen libraries of lots of drugs and molecules to find things which stop them binding, so fit in between them and stop them touching each other. And then we can go to the lab and test these drugs in the lab and see if they actually work. So if you can see here in green, this is actually one of the molecules that I'm looking at the moment. And when we look down the microscope, you can see that the fungi without the drug grows as this mycelial mass, but when we add this drug, they're not be able to grow as well. So another project that we have going on at MFIG is the YAC project. So when these fungi invade our cells, one of the main mechanisms they can do this is by the stress response. And this is the fungi building these cross walls called septa in between their long strands of hyphae. So you can see here in this diagram, they're building these walls, which is called the stress response. And this closes off compartments of the fungi so they can keep themselves safe from our human defense mechanisms attacking them. So one of the proteins that allows them to do this is called the YAC-A protein. So this protein is really important to be able to shut themselves off from human attack. So we think if we could stop this protein from functioning somehow, it'll be easier for our cells to attack the fungi, and this could slow down the infection and try and stop it from penetrating into our cells. So we're doing a similar things to the NIM project with this. So we're looking at the shape of the YAC-A protein and seeing if we can find molecules that stop it working somehow. So yeah, just to summarize, the things that we're doing in the lab is we're using a lot of computational modeling to look at the shape and how these proteins interact with each other, and then finding molecules that can stop them working. And so using the computer modeling, we can screen hundreds of thousands of things all at the same time, which saves us time before going into the lab and seeing if they actually work. And we can use chemistry to make existing drugs better, so making them a bit less toxic to us, making them try and overcome resistance by changing the drug's shape and structure a little bit. So this can help us save what we already have. And as Kaylee was talking about before, we can use new models to assess if these drugs can work. So the first preliminary steps in the lab, if we find something interesting, we can test these on new cell models, which are cheaper and easier and quicker. So thank you all for listening, and I'll take any questions if you have any.
That was really good. Thank you, Izzy. Did we get any questions?
Yeah, we've got one in the chat that says, can we use YAC-A as a drug target to treat candidiasis?
So yeah, that's really interesting. I'll need to talk to Sam because he's the main person that works on this project to see if Candida and Aspergillus have this protein in common. But yeah, if they both have the same YAC-A protein and it's a similar shape and is the same across these fungi, then yeah, we could potentially use anything that we may find in the lab to see if it works against both types of fungi.
That's brilliant. Thank you, Kaylee.
Yeah, I just wondered, looking at that, where does the investigation begin? Does it begin with the target that you're interested in, or does it begin with the drug that you have available to test?
Yeah, so there's multiple different starting points that you can take. So for example, with the NIM project, we knew that these two proteins were essential for the fungi to grow, and then we used the protein as the starting point. But I think for the YAC project, it's the other way around. So we used the molecule as a starting project, because some of our collaborators in Canada, they've been looking at this protein in a different organism, and then we saw if that drug could work with Aspergillus. So you can kind of start it from either end of the spectrum, and yeah, multiple approaches.